啊，我这两天正在读一本，嗯讲天才科学家的一个书，就是讲 NIH 的 oral history。然后
那以
你
前，
会发现，这
前
个在50、60年代的 NIH 是多么 exciting 的一个地方。
哎呀，不要提伤心事了，不要提伤心事了。
嗯，但是这个都是你看那个在50、60年代 NIH 是很 exciting 的嘛，它直到它刚刚建立20、30年代是美国所有的大学都嘲笑是一个养老的地方，所以总是有30、40年河东40年河西的嘛，不用太。
哎，是，我正好赶上河西这一段时间。对，河东的时候我没赶上，我全赶上河西的时间了。
那你就做中流砥柱吧。
哎呦，行了，我不被淹死不错了。
嗯
我们差不多开始好不好，是吧
，OK，好
？Okay, so let's get started. Um, Hello everyone, welcome to our today's museum seminar. It's really excited to have my old friend, also colleague, Dr. Liu Zhihua from Tsinghua University, being with us today to share some of her recent work. So I think I know Zhihua now for over decades, probably more. So Zhihua currently is associate professor um, at School of Basic Medical Sciences at Tsinghua University. She finished her undergraduate training in biotechnology in Beijing University, and then continue with PhD in biochemistry and pharmacology in Harvard, uh, in USA. So after the PhD training, she continued with her postdoc training in National Institute of Health from 2006. And then she, from 2012, after finish postdoc training, she moved back to China, start her independent research program as a PI in Institute of Biophysics, Chinese Academy of Science. And then afterwards, from 2020, she relocated into Tsinghua University as associate professor, very much until today. So I think probably a lot of us in the audience are familiar with Zhihua's work. So her lab primarily interested in studying the interactions between symbiotic microbiome and their host. And then her lab really achieved innovative research outcomes in the field of microbiology and immunology. Her work has reviewed a new paradigm of host microbial interaction, particular the gut-derived lysosome-released bacterial products, which are recognized by intracellular innate immune receptors in multiple host cell types, regulating cellular secretory activities through a unique genetic pathway. The pathway promotes intestinal homeostasis in cell and has also been identified in pancreatic beta cells, where it contributes to maintaining blood glucose homeostasis. These studies actually really deepen our understanding of host microbial interaction and provide important insights for exploring the relationship between gut microbiome and the human health, which I think she's probably gonna to tackle this topic quite a lot in her today's talk. And then Zhihua really made a lot of groundbreaking discoveries. And then with these, she's received multiple awards, which including a Young Investigator Award from CBIS Science, China Life Sciences, and then excellent teaching award from School of Basic Medical Sciences from Tsinghua University. So it's really a great pleasure to have you, Zhihua, with us today, and then we are very much looking forward to your talk.
thank you, Tran, for your very flattering introduction. It's very long. Actually, I didn't expect you give such a long introduction, but it's very, very heartwarming. And it's my great pleasure to be here again today. And, mostly I participate in the Immune Zone meeting a lot, and I see amazing talks from all my colleagues, and, a lot of thanks to our founding organizer Chuan and many other organizers, for example, Li Hao and Xu Zhou, etc. So today my talk, the talk title of my talk is a bit unusual. So actually, I apologize for this very unusual title because actually I like it, but it's confusing because how the host crosstalk to the mitochondria, right? So I chose this title because actually it kind of reflects my research line in my lab. So today I will mainly present two of research line in my lab. One first is my initial what Chuan has talked about. We focused on the host and microbe crosstalk mediated by lysozyme action on commensal microbes. And the other line comes from my research, actually. When I think about how the action of the lysozyme on the microbes and host, then I started to think how the mitochondria people has long been hypothesized it is so-called endocytosis, the microbes. Then I started to think maybe mitochondria may also mediate cell-cell crosstalk in a similar fashion. So that's why I come up with this very unusual title. All right, so as we all know now that actually commensal microbes play very diverse a diverse role in our host by very conventional, including helping digestion, pathogen. Okay. Do you see the laser line? No, this is not a laser line. Okay. No.
If you want to choose the laser line, you can go to the left lower corner. I think there's
it says I should allow it. Okay.
I think the arrow point is okay.
All right. Can you see the arrow point?
Yes.
See my mouse.
Yeah, I can see that. Yeah, I can
Yes,
see your.
okay.
Yeah.
All right. So all the immune training, but for unconventional role of the commensal bacteria, it has been more complexing or more mysterious thing is, for example, stress response. For a long time, people have shown that actually the dysbiosis somehow affects the stress response or other things. For example, the neural development and metabolisms. Actually, my lab work has been starting to provide some evidence how the microbes may modulate this response. So my work all my lab started being focused interest from the inflammatory bowel disease. Actually, not, every people for most people probably in this seminar would know that actually. Globally, we have seen the increase of inflammatory bowel disease, and actually, inflammatory bowel disease has many factors. One of the many factors is the genetics. Genetics plays a quite big role in the development, possibility of development of IBD, and among them, our lab has particularly interested in a group of cells in the small intestines called Pani cells. And actually, a number of genes, genetic susceptible genes associated with IBD, actually they have all specific phenotype affecting the Pani cells. And to us, one thing, particularly for interest in our Pani cells, is actually Pani cells secretes many antimicrobial peptides, which are able to modulate the gut microbiota and also, keep a distance from the microbiota away from the epithelium, right? That's what we understand how the Pani cells helping building the intestinal intestinal barrier. My lab was particularly interested in how the lysozyme in the penis cell is secreted. So in our first line of research, we found actually the penis cells secreting lysozyme in a highly regulated pathway. It requires the bacterial peptide, mirror dipeptide from the commensal bacteria to be sensed by NOD2 receptor in the penis cells and assemble a sorting complex in the dense vesicles in penis cells. Without this complex, the lysozyme would be sent to lysosome for degradation. So after this long research, I was thinking, why the lysozyme need to undergo such straightened regulation compared to other antimicrobial peptides in penis cells. Therefore, we thinking that compared to other antimicrobial peptides, lysozyme actually is an enzyme. It's in charge of concleaves the peptidoglycans on the cell wall component, cell wall, and potentially releasing the so-called PAM molecules from the cell wall, and which can getting absorbed in our body and get into our cells. So actually, people already start to show that actually the NOD1 ligand as well as the NOD2 ligand either in neutrophils or in the macrophage. Actually, some of them, the ligand was from our commensal bacteria. And actually, the engagement of this pathway actually responding to the commensal bacteria is in charge of many factors, many processes. For example, the lymphoid tissue genesis. Also, for example, even they can get into the brain to regulate the body temperature central stat. And thinking that to be very naive, we generated a lysozyme 1 knockout mice, and we found that in the mice without lysozyme 1, with the penis cells stop generating lysozyme. Of course, the mice has another allele called another gene called lysozyme 2, but by doing lysozyme 1 knockout, it depleted the lysozyme in the intestinal lumen, and then we use the report assay to see whether the depletion of lysozyme 1 in penis cells would affect the NOD1 and NOD2 ligand in the circulation. So here we see that in the Y-type mice with SPF condition, we can see a level of NOD1, NOD2 activity and NOD1 activity. In the the germ-free mice, we can see a significant drop of the NOD2 ligand and NOD2 level as well as NOD1. And if we lysozyme 1 is depleted, the level of NOD2 and NOD1 is significantly reduced. Therefore, it's consistent with our hypothesis. Actually, in the most conditions, if the intestinal lumen lacking the lysozyme, the level of NOD1 and NOD2 ligand into the circulation is greatly depleted. Of course, people start to realize actually some, especially some probiotics bacteria, actually can actively secrete NOD2 ligand into the environment. Therefore, it's more, maybe it's also represent a way how those probiotics can enhance the crosstalk between the microbe and the host. Here, we show that. Yeah, here, sorry. Here is the insulin the pancreas isolated beta cell study, we found, as we could understand that actually the beta cell secretes insulin, but it has a form of precursor form called proinsulin. Usually, proinsulin is restricted in the Golgi area. After processing into insulin, it goes out. Therefore, we can see a very nice spatial organization of proinsulin to insulin. However, we found that this process was disrupted in the germ-free mice. Okay, so in the germ-free mice, the proinsulin and insulin no longer segregates, and this is not a developmental problem associated with being germ-free, because if we put the germ-free mice into the environment for 1 week, we can see actually the spatial segregation of a proinsulin and insulin is restored. And here we found actually this commensal bacteria-guided insulin trafficking actually depends on the lysozyme signal. Lysozyme depends on the lysozyme because if we knock out the lysozyme 1, we pretty much recapitulate what we see in the germ-free mice. And through the molecular delineation, we found actually it was the beta cell. Its cell express the NOD1 molecules, and the NOD1 molecules play a cell intrinsic role in beta cells to guide the insulin processing in beta cells. Okay. All right, so here is our summary slide, and we have, in addition to the Paneth cells, which we show that lysozyme, by digesting bacterial wall to generate NOD2 ligand to itself, actually, it's regulating lysozyme secretion, and we have beta cells in the final. This line we have our last final part is on the adrenal gland chromaffin cells. We found actually the signal from the commensal bacteria here is NOD1 again, and helping the epinephrine packing or storage in the secreting granules. Without the commensal bacteria, the amount of epinephrine stored in the secreting granule is almost only half of the what is stored in the SPF mice. Therefore, the stress response in the mice was greatly reduced. It's like well, the bacteria is helping us to handling the stressful environment. So here we like to all summarize our research. We show that actually lysozyme, in addition to its role, maybe modulating the composition of commensal bacteria, actually it's enzymatically releasing the peptidoglycans from the bacterial wall, and releasing small molecules getting to our circulation and to communicate with different cells in our body to helping our body to better adjust to our environment. So people has been very long talking about maybe in the very, very, very ancient time, one cell endocytosis the aerobic bacteria, and the aerobic bacteria decided to stay inside one cell and become a mitochondria. And in fact, mitochondria has been so much integrated part of our cell, because it becomes our central powerhouse to generate energy for our cell, and also it governs the cell destiny by being a crucial part of cell death pathway. Also, because actually it contains the mitochondrial DNA. Actually, people have realized that it could initiate the immune action activation. And lately, there's a lot of study try to found actually a very exciting part about mitochondrial life. For example, yesterday, Xinguo Liu in the Shenzhen has just shown that if you transfer mitochondria into mitochondrial failing cells, actually it can play a very positive role. So this is part of a scenario of mitochondria for the intracellular exchange. Okay, but my question is, since actually mitochondria is such essential of cell health, right? Because when mitochondria is oxidized, it's need to degraded. If it's not well controlled, cell go under cell death or immune activation. Therefore, I'm thinking that maybe before cell go all these pathways, going cell death or going immune activation, maybe they have a way to able to communicate with the surroundings, right? So, what I mean is how much the mitochondria is activated, how it gets the status communicated to the outside environment, especially the macrophages. So I started to think about it, and then how macrophages come into the thing is we always talk about the afrocytosis. All every day, like we have millions of cells undergoing cell death every day. But yet, in a healthy body like us, or when we're very young, a particular body can never really absorb. So one thing is first the afrocytosis helps clean the debris. So it does not triggering the inflammation, right? But the second, people starting, actually a long time ago, people know that afrocytosis exert tissue repairing effect on the tissues. But lately, people even become more realize that afrocytosis actually is not a silent process. Actually, it's a active process. It can exert a profound effect on the recipient cells to remodeling the, so for example, cell proliferation, the macrophage renewal, as well as the metabolisms. So I'm thinking that maybe the afrocytosis the macrophage could respond to the cells mitochondrial activities in other cells. So here come with my collaborator Li Yu in Tsinghua University. In 2021, he published a paper that showing that the cell can excrete the damaged mitochondria through a process called mitocytosis, in which actually the damaged mitochondria get packaged into the secreting vesicles. Here we call the mitosome. And I was wondering what happened to the mitosome? Can it mediate a crosstalk between the host of donor cells and recipient cells? So thinking about mitochondria, so I'm obviously would turn my eye onto a tissue called brown adipose tissue, because actually that's a cell contains highest density of mitochondria, right? So the brown adipose tissue is a tissue responsible for our heat generating process. When we encounter the coldness, our hypothalamus get activated. It sends out the somatic nerves activation to trigger our muscle to shuffle, and at the same time, the sympathetic nerves to activate our fat tissues or the binge tissues to start a thermogenesis. The thermogenesis, of course, the highly depend on the decoupling activity of mitochondria. And compared to the regular white adipose tissues, the brown adipose tissue or binge active tissue contains highly innervated vessels, also the nerve fibers. And actually, many studies have shown that upon the coldness, upon the cold exposure, the sympathetic nerve activated, and consequentially, it activates the brown adipose tissues, which activates the mitochondria and the decoupling process, and the mitochondria get really crazy to generate heat. And at the same time, the blood vessel get activated and bring out the circulating blood and bring out the heat generated. That's how our body temperature was maintained. Indeed, in humans, study has shown that upon the cold, we can see huge surge of the blood supply into our BAT tissues. Okay. My question is, when the brown adipocytes is generating heat, it turn up the mitochondrial activity. I was wondering, it would very much make sense that if the mitochondrial activity in the brown adipocytes could couple with the blood supply, right? Logically, it makes sense. In addition to the activation of synaptic nerves to regulate the vasodilation, the mitochondrial activity should be also a final tune for the blood supply. So here we use the mice which Lee has used for the mitocytosis. We found actually the T-span nanocow mice had a defect in maintaining the body temperature upon coldness. Okay, here we found that even at room temperature, the mice has a lower body temperature, and it's exacerbated at 4 degrees. And consistently, we found that brown BAT, the bats, has more lipid accumulated in the BAT. Okay. And we found the similar thing. So if we depleted the T9 in adipose tissues, adipocytes, using the UCP1 driven promoter, driven Cree, and we can see that it cannot resist to the cold. And if we deplete the T9 in myeloid compartment, and the mice was pretty much behave with the T9 knockout mice, T9 flox control mice. Therefore, we decided that we concluded that T9 is required in the brown adipocytes for the homogenesis. Okay. So when starting to think whether this the dependence on T9 also involved in the mitochondrial secretion. So here is primary cultured the brown adipocytes, and we found that if we stimulate the cells, we can see actually a number of mitochondria start to appear on the cell surface. On the cell surface, the TIM23 marks the mitochondria. And if we do the real-time imaging, we can see actually the mitochondria is getting out of the brown adipocytes. And through a complicated fractionation, we found actually from the brown adipocytes, see we able to purify the extracellular vesicles containing the mitochondria, pretty much similar to what Lee has published in 2021. And so it has a typical size of 900 nanometers, very typical of the microsomes. And it actually not only compared to other vesicles containing mitochondrial fragments, this mitochondrial fragment, this mitochondrial vesicles containing both the outer membrane vesicle as well as the intermembrane vesicle. It's also from the structure we can tell it's more like a complete, it's a whole mitochondria, but it's damaged mitochondria, and we compared the Y type and the T9 knockout mice, and we found compared to the Y type mice, which generates a lot of mitochondrial vesicles, the T9 upon cold exposure, it cannot generate similar amount of the mitosomes. And we also quantified the number of the mitosomes generated by the Y-type mice or the T9 knockout mice, and pretty much we see what we can, visualize in the electron microscope. Therefore, here we reach our half conclusion that upon cold exposure, the brown adipocytes actually there's a lot of action in the mitochondria, and inevitably some mitochondria will undergo damage. And we found that upon the cold exposure, the mitochondria, the brown adipocytes actually expels some mitochondria in a process called mitocytosis, and in a vesicle called mitosome, and it's T9 dependent. So we next want to ask whether a macrophage would respond to this process. So we add the mitosomes into bone marrow macrophage, and we can see that very quickly, without surprising. Here we have a lot of immunologists here, so it does not take a genius to know that actually macrophage will happily take up whatever debris in the cultures, right? And that's exactly what we see. And we found actually, we also did a really simple experiment to put the vesicles onto the macrophage, and also in comparison treat the macrophage with the interleukin-4 and IPS, and we found that the treatment of the mitosome makes the macrophage halfway, between the M1 differentiation and M2 differentiation. Okay, so we think maybe we should look at what happened inside of the mice. Therefore, we first giving the mice cold exposure and performed a single-cell RNA-seq on the mice, and we found the macrophage actually in the mice can be separated into the 4 populations. Is class 1, 2, 3, 4, and actually the class 1 and 0 and 1 more represents the M2 polarized macrophage, and the 2 and 3 are more inflammatory macrophage. We started to see whether the mice, the T1 mice, T9 mice, and Y-type mice would have different population of macrophage upon coldness. And we found when the mice are exposed to cold, the M2 macrophage in T9 knockout mice was drastically reduced, and therefore here. One more thing we noticed, actually, because this population is enriched in the cold exposed Y-type mice, and these mice also this population has a cell receptor called MOT-TK. Actually, MOT-TK families are major families involved in the efferocytosis. Therefore, we start to think maybe that's a way how macrophage, and especially the M2 macrophage, responds to the mitosomes. Therefore, here we get the MOT-TK knockout mice, and here we use the perennial macrophage because actually bone marrow macrophage does not express high level of MOT-TK. So, but peritoneal macrophage express high level of MOT-TK. And we found that if we compare the wild type and the MOT-TK knockout macrophage, we found that the process of uptaking the mitosome was greatly reduced in the MOT-TK knockout mice. Okay, so it apparently MOT-TK is a receptor mediates the uptake of the mitosome, and we also want to know whether the engagement of mitosome with MOT-TK triggers the signaling of MOT-TK. And indeed, we found actually the mitosome activates the MOT-TK. And we want to see whether this leads to activation in vivo. Here, we did a immunostaining for the macrophage, which we also in the single cell data, we know that this macrophage, the class 1 macrophage, class 0 macrophage has the express LYVE on the surface, means represent its macrophage very close to the blood vessel. And we found that in a wild type mice, upon the cold exposure, the MOT-TK is activated in the LYVE positive macrophage, while the MOT-TK knockout mice, we don't see the signal. Okay, so we start to focus on the MOT-TK now. And we subject the MOT-TK mice to the cold, and the mice 2 hours, as we expected that mice, similar to the T9 knockout mice, does not is sensitive to the cold exposure. And here is one thing very striking here. So the mice actually is sensitive to cold, and here is the infrared body temperature. We can show that we can see that in the here we can focus our eye here, right? Here is what the brown adipocyte, brown tissue, yes. And if you remember our first image showing the T9 knockout mice, the mice is all yellow. However, in the MOT-TK mice, we see that even though the overall temperature is lower, actually the temperature around the brown adipocytes, the brown fat was intact. So it means that actually in the MOT-TK mice, even though the whole body temperature is reduced, however, the local temperature in the brown adipose tissues was maintained. And extremely surprising is if we get the brown tissues from the mice, this is in sharp contrast from all the phenotype we see before. We see that actually MOT-TK, in the MOT-TK, the BAT, the lipid is completely exhausted, right? Otherwise, we would expect there's a lot more droplets, right? But here we see that almost completely depleted lipid droplets in the MOT-TK. And if we use the laser spectral microscope, we can see that in the wild type mice, upon cold exposure, we see highly upregulated blood circulation in the brown tissues. However, we can see at the 4-degree, the MOT-TK has the blood infusion is greatly reduced. Okay. Therefore, oops, sorry. Therefore, think we think there's some problem with the mice that's very different from the whole body knockout mice. Actually, the brown adipocytes is intact because actually the brown adipocytes does not express MERTK at all, right? But therefore, it has no problem of generating heat, right? But the lack of MERTK in macrophage somehow is causing the problem, right? And we also see the reduced blood supply. Therefore, we go back to examine how the blood supply is regulated during the cold exposure. To our surprise, all the study has been completed before 2004 because ever since this ground review of this bone marrow, also no, brown bat physiology, no people ever studied how the blood supply is regulated upon coldness. So in this 2004 review, this shows that this model summarized is NE activates the brown adipocytes and generates and somehow regulates the NOS and generate nitric oxide, which dilates the blood vessel. Ever since, no other study has looked upon this problem. Until 2023, there's a beautiful metabolic influx study published in the Nature Metabolism. This shows that within 30 minutes of cold supply, they extract the BAT from the mice before and after the coldness. They didn't extract. They take the blood out of the BAT before and after the cold exposure, and they measured the difference between of the metabolites in the blood. And this what one of the most striking most dramatic change in metabolite is prostaglandin 1 E1. Actually, they see a huge production of prostaglandin E1 upon the coldness, and we pretty much replicate what their phenotype, what their publication in the white mice within the 30 minutes with a short time of exposure. We can see a huge surge in the PGE1, but the increase in the PGE1 was abolished in the MERTK knockout mice, and we want to know whether the failure of production the PGE1 was the problem. Therefore, we supplement the mice with PGE1, and then we reproduced pretty much the result. We have shown that actually, when giving the mice PGE1, the MERTK mice no longer cold sensitive, and actually also the circulation through it restored the circulation to the peripheral, and the lipid droplets depletion is also suppressed. So here is our final model. Actually, when the brown adipocytes get activated, some damaged mitochondria get out from the brown adipocytes, and the macrophage nearby is uptaking the mitosome through the MERTK-dependent receptor, and it generates the PGE1 from the uptaking cargos. And the PGE1 is, people already know for a very long time, even used in clinics, that actually PGE1 able to increase the blood supply to bring the oxygen, glucose, and fat from the peripheral to fuel the mitochondria. That's the reason why multi-key knockout mice actually after cold exposure, it completely depletes the lipid droplet. And also very importantly, the blood supply is required to bringing out the heat generated from the mitochondria in the brown adipocytes. So here is our, another level of summary that in the traditional view that sympathetic nerve inactivation turn up the blood vessel. That's what we see also in the multi-tick knockout mice. We see even without multi-tick, we still see the level of increased blood supply. But we think on the top of the synaptic nerve activation, mitosome being secreted from the activated mitochondria acts as the immune metabolic messages, which get able to directly connect or link the mitochondrial activation in the brown adipose tissues with the blood vessel dilation. So it's more like a local thermostat in some sense. So here is our final summary. And we think that mitochondria, in addition to cell intrinsic being playing a role for the power generating, actually part of them can be a signal sending out to the environment, getting talked to the macrophage, and the macrophage maybe after responding to this signal, able to find some way to contribute back. And of course, the PGE1 is maybe one of the many signals sending back to the cells, donor cells, which we of course are actively looking for the other, maybe there are other contributions as well. So finally, I'd like to thank my lab members. This work actually mainly was conducted by Yabo Zhao, as well as the students. Jia Yi contributed a lot. And also, I'd like to thank my collaborator, especially Liu, Igor Wang, and Wang Qiang, who helped us for the microsome purification, also the multi-tick knockout mice, and initial the brown adipocytes culture. And also I thank my my collaborator and dear colleagues in Peking University, Yi Qiufu, for helping. Actually he helped a lot, especially we haven't been done a lot of work before in the brown adipose tissues. And finally, I'd like to thank my founding agent, and also I'd like to thank all of you for your listening for my talk and for your time.
Okay, thank you so much, Zhihua. I think it's really interesting study. We can start the question session. Anyone questions, please raise your hands or put your question in the chat box. Yikui, go ahead.
Hi, thank you. Thank you, Professor Liu. So I think brown adipose tissue are quite important for our health, right? So I mean, in adult, we have fewer such kind of tissues. Is that possible? That how to increase the brown adipose tissue in adults? Is that possible? We can do that through as you mentioned, host to microbiota crosstalk. This is my first question.
Host microbiota crosstalk. Yes, actually, I think just a few days there's a very nice publication showing that actually, I remember it's bioacid. Some parts of bioacid could participate in the brown adipose tissue genesis. And actually,
cool.
one more problem, one thing, one issue we are hoping next, to develop this process, because actually you can see I didn't irritate how the macrophage perceives mitosome, right? Actually, we found actually there's a array of signaling pathway after get activated after receiving the mitosome. So we are also trying to see whether the mitosome could helping the macrophage to giving more signal to the pre-adipocytes to differentiating to the brown adipocytes. So that's a possibility as well.
Yeah, that's very interesting. Thank you. And my second question is that, there are a lot of microbiota in the gut, right? So definitely there are a lot of mitochondria in bacteria. So is there any, mitochondrial transportation or crosstalk between these two cells, so our host cells and the bacteria cells in the gut?
Oh,
Direct, transportation between the host and the bacteria in terms of mitochondrial transportation.
oh no, I didn't. I'm not aware of it.
Okay. Yeah, thank you so much. Because,
Yes,
so right now, the mitochondria is so important. So there are a lot of mitochondria transplantation therapies, right? So
yes,
I'm
yes.
wondering, since we have so many bacteria in the gut, whether there's any transportation between, so can we just steal or just grab some healthy mitochondria directly from the bacteria in the gut so that to keep our, digestion system more healthier?
That's that's interesting. Yeah, yeah. I don't. I'm not aware of it, but
Oh,
definitely very interesting.
yeah. So the following question is that is there a difference in mitochondria between the host human and bacteria?
Oh.
Are they the same or is they are functionally or structurally different? I'm not sure because originally they come from the same ancestor, right?
Actually, I think I didn't it's you asked them that totally in my die zone, so I don't know. But according to Doug Green, that actually even inside our cells there are two types of mitochondria. So it's
Uh-huh.
we have to say that we tend to it's like error which we look at the microbiota as a piece of shit. Of course there are, and but the whole piece. Nowadays, people realize actually the microbiota has actually they are having so much diverse role. So I think people are on the edge of understanding the secret life of mitochondria, right? They also has very diverse. I always wondering whether the mitochondria in different cell types or even different. But in the different organs could be very different. Indeed, we
Mhm.
can say that actually at after some stimulation, especially the macrophage undergoing apoptosis, actually the mitochondria are very different when compared to what they are before the apoptosis, and they are secreted into different groups. And of course, it's new tools is definitely needed to answer this question.
Thank you so much.
There's a question from the audience, Xiaoqing, Wei Xiaoqing. The question is, does efferocytosis of mitosomes by macrophage induce a M1-like or M2-like phenotype? Also, what mechanism underlying cold-induced mitosome formation in adipocytes, and how does the release of damaged mitochondria components from the mitosome drive PG1 production in macrophage?
That's a great question. So we know that, of course, M1, M2 is very, fake terms. And actually, we can see that efferocytosis taking up by mitophage through the taking up the mitosome by macrophage actually drives the M2 like phenotype. But in body, in the cell, in the mice, it's a class 0 macrophage is more expanded. And the mechanism underlying the code induced the mitosome formation actually is an excellent question. I didn't put data here. We show actually it is dependent this process is very complicated. It looks like now we show actually it's a microsome dependent pathway, and we also have data. We show here is T9 dependent, and it's a microsome like structure. And actually, we don't put data here. Actually, it depends on the autophagy. It depends autophagy pathway uptaking the damaged mitochondria. So it's two pathway connected, linked together. And this is another excellent question. So how does the release of damaged mitochondrial components of mitosome drive PGE1 production? Okay, so first of all, it actually took us a very long time to realize. Actually, it took a very long time for us to understand why the PGE is in this process. Because, to be immunologist, people already know that if you need to produce PGE, we need a COX-2, right? And actually, the COX-2 needs a long time to produce, right? Because it needs NF-kappa B12 activated to drive the production and the protein translation of the COX-2. And in fact, in the mice, the cold process is a very fast process. We cannot wait for 2 hours or maybe 30 minutes for the cells to generate COX-2, right? So until we realized in this scenario, it's not the COX-2. We know now it's COX-1 in the macrophage is activated, so it's not translated or protein transcribed. It's actually direct activation of COX-1 in macrophage. Actually, the material inside the mitochondria when get into the phagosome, get digested, and get transported out from the phagosome and get processed by COX-1 to generate the PGE1.
Okay. Shu, go ahead.
Hey, Zhihua, really, really cool work. Really good to see you here. So fascinating story for the mitochondria. You mentioned that the bone marrow macrophages do not have high expression for MERTK.
Yes.
And do they also uptake these vesicle structures that you isolated?
Yes, it's actually, yeah. Macrophage has different process, ways or mechanism to uptaking things. That's actually we always use bone marrow macrophage, but actually the problem is initially we see that Y type and multi-TK bone marrow macrophage in uptaking of mitosome does not have many, the difference is not big. So then I think maybe that bone marrow macrophage does not express the multi-TK at sufficient high level. That's why we switch it to the peritoneal macrophage.
I see. And it sounds like since you see the phenotype drastically so quickly within 4 hours in the cold challenge, I wonder if there are differences at baseline in the adipose tissues, in the white adipose tissues, in the either the MERTK knockout mice or the T-spin9 knockout mice. Have you looked into potential innovations in the fat tissues, if that may play a potential role in driving the cold challenge particular phenotype?
This is an excellent question. So when the mice was maintained in room temperature, actually it's a condition actually still neutral, temperature neutral compare for the mice, right? So indeed, actually in the T9 knockout mice at room temperature, we can see there's more lipid droplets accumulating. Therefore, it indeed could indicating there's a baseline difference, right?
That's in the brown adipose tissue.
Yes.
It
Yes,
also happens in the
more
white.
triplets.
Right,
There's
right. But I thought the data you showed there that the main differences are probably the white adipose tissue driven thermogenesis during the cold challenge that may be different between your wild type mice and then the vertically knockout mice.
Oh no, we used the cold acute
Right.
cold exposure. So actually the mice at white type and Q, we don't see the difference in the white adipose tissues. So therefore we focused on the brown adipocytes.
I see. Maybe I
And
misunderstood your data.
Also the baseline level, we don't see the macrophage difference.
Okay. So it's really code-induced vesicle release that driving PGE2 feedback in a process that facilitating thermogenesis in the brown adipose tissue.
Actually, therefore, we didn't put in data on the T9 knockout mice because that mice, originally it already has the lipid droplets accumulating in the brown adipocytes, around the sites. Therefore, we used the multi-decay mice. Actually, the multi-decay mice at room temperature was pretty much similar with the white type mice.
Very cool, very cool. And maybe one question, maybe touch on by the last question is that, what do you think would you think is primarily metabolic driven in the way that the content of mitochondria are released within macrophage are driving PGE2, PGE1 access, or there are internal sensing pathways similar in the analogy you made from bacteria to hosts in this case mitochondria across different cells that may be being sensed in some unique way are modulating the downstream process. Either is entirely metabolic driven, or is potentially there are unique ways sensing the being transferred mitochondria to drive that process.
That's a terrific question. That's exactly we are, pounding on it. So we are trying to generalize the lipid content of the mitosomes to see whether the lipid contents is the determining factor to drive the production of PGE1, not the PGE2. So this, to us, it's very surprising that in our experiment, as well as a paper published in 2023, showing that the PGE1 was the major player, the most changed prostaglandin in the BATs. And we don't see the change in PGE2 at all, between white type and Q. And from the before the code and after code, it's only twofold change in the level of PGE2. Actually, the PGE1 level change is more than 20-fold. So somehow there's a specificity in generating PGE1. So we don't know why is PGE1 specifically generated here rather than PGE2. So we don't know.
Very cool. Yeah, thank you.
Thank you. Very insightful question.
So I have a question after Xu's question. So the Merck TK macrophage, is this a generic phenotype in the peripheral tissue resident macrophage, or their expression pattern is different from tissue to tissue?
No, I didn't examine it. That's
Okay,
a good question. So MUT-TK, I know the expression pattern here we observed around the vessel location, the LYVE positive express high level of MUT-TK. It's true in the cardiovascular system as well, but we don't know, generally speaking, in other tissues.
Okay. And then because we know that brown adipocyte to generate heat, there's different way to do that. I mean, to utilize UCP1 is one way, and then obviously to generate PGE to dilate blood vessel is another. So my question is, if you deplete the macrophage and induce cold exposure, will you still see this difference?
People has did it. People already did it. So that's the reason we use the multi-TKKO to do it.
I see. And then